Hello friends, welcome to the online education. This is the lecture number 3 and subject is physical chemistry and analytical chemistry. So we have studied about the platinization of electrodes. First of all, what is the platinization of electrodes? So first of all, electrodes are coated with the divided platinum black. It's called platinized electrodes. See the figure or imagine the figure. Both are platinum electrode and it is coated with the platinum black. So it is called platinum electrode. The electrodes are coated with divided platinum black. So it is called platinized electrodes. And this process is called platinization of electrode. So this is the definition of the platinization of electrodes. Electrodes are coated with the divided platinum black. It's called platinized electrode and this process is called platinization of electrodes. First of all, platinum electrode of conductance cells is cleaned by the chromic acid or distilled water. First of all, platinum electrode. Two electrodes. Platinum electrodes are cleaned by the chromic acid and distilled water. And then the platinization is carried out by taking 2 to 3% solution of chloroplatinic acid. Chloroplatinic acid means PtCl4. First of all, platinum electrode of conductance cell is cleaned by the chromic acid and distilled water. Then after, platinization is carried out by taking the 2 to 3% solution of chloroplatinic acid. This is the chloroplatinic acid and this concentration is 2 to 3% concentration. So see the figure. Take a beaker and into the 2 to 3% concentration solution of chloroplatinic acid and two platinum electrodes dipped into the beaker in the solution and it is connected with the rheostat. This is the two-way switch rheostat. This is the two-way switch rheostat. See in the figure. Now 0.0 to 0.03 gram at lead acetate in the cell. Lead acetate also added a lead acetate. 0.0 to 0.03. 0.0 to 0.03 gram add the lead acetate in the cell. Now for this process, conductance cell is connected to a rheostat. So the conductance cell, this cell is connected to the rheostat. Source of current and two-way switch is to connected to a rheostat. Rheostat is connected with the two-way switch. See in the figure. Now pass the 4-volt current. Both sides pass the 4-volt current. Pass the 4-volt current from the source about 15 to 20 minutes. The current is passed from 15 to 20 minutes from the cell. Due to this electrolysis of PtCl4, means chloroplatinic acid. It occurs and platinum electrode of conductance cell is platinized. And then after this process, the conductance cells is platinized. So this conductance cell is platinized and this process is called platinization of electrode and the conductance cell is platinized. Now this process is repeated, pass the current and form the gas bubbles. Pass the current, then after gas bubbles are see in the our solution. Every 30 seconds pass current. Every 30 seconds pass the current from the two-way switch, and it is connected with the rheostat, and rheostat is connected with the two electrodes, two platinum electrodes, and two platinum electrodes are dipped into the solution of the PTCl4, means chloroplatinic acid and lead acetate solution, and chloroplatinic acid solution concentration is 2 to 3%, and lead acetate will be added 0.0 to 0.03 gram in the solution. Now the electrodes of the conductance cell is platinized. Then after the process is completed and the electrode are platinized, and this process is called platinization of electrode. And then after reaction under the cell is the PTCl4 gives to Pt plus 4 plus chloride ion. This process is called ionization. Then after 4 mole of chloride ion gives to the 2Cl2 plus 4 mole of electron. This process is called oxidation. And then after Pt plus 4 plus 4 electron gives to platinum metal, and this is process is reduction. So this is the platinization of the electrode. So this is the concept of the platinization of the electrode. First of all, we take the beaker and dip into the solution to platinum electrode. The chloroplatinic acid solution has a 2 to 3 concentration, and then after we added the 0.0 to 0.03 gram lead acetate, and two platinum electrodes are connected to the rheostat, and rheostat it will be connected to the two-way switch, and then after we will pass the 4-volt current, and it is the 15 to 20 minutes 4-volt current passes in the cell, and then after the cell is platinized and the conductance cell is platinized, and this process is called platinization of electrode. So electrodes are coated with the divided platinum black, it's called platinized electrode, and this process is called the platinization of the electrodes. So we will study about the platinization of the electrode. Now we have studied about the neutralization titration. Neutralization titration have 4 types, and we will study about our first titration method, strong acid with strong base. We will study about the neutralization titration of strong acid with strong base. This is our first titration method, strong acid with strong base. So first of all, strong acid with strong base titration considered as the titration of the HCl with NaOH. HCl means hydrochloric acid and NaOH means sodium hydroxide. HCl is a strong acid and NaOH, sodium hydroxide, is a strong base. And the reaction was HCl plus NaOH gives to NaCl plus H2O. This is the titration process. HCl plus NaOH gives to the NaCl plus H2O, and also called this is the neutralization process. So this is the strong acid with the strong base titration considered as the titration of HCl with NaOH. Hydrochloric acid is a strong acid and sodium hydroxide is a strong base. Acid and base both are strong electrolyte. Acid and base both are strong electrolyte. So it will be participated to the Na plus plus Cl minus will be ionized. Therefore, they are very fast and completely ionized because they are strong electrolyte. So acid and base means hydrochloric acid and sodium hydroxide both are strong electrolyte. So it will be very fast and completely ionized, and the ionic reaction is given as under. So HCl means 2 ions, H plus plus Cl minus, and NaOH, sodium hydroxide, Na plus plus OH minus gives to Na plus plus Cl minus plus H2O. This is the water molecule, and this value are lambda infinitive value, the conductance definite lambda infinitive value, dilution value. So H plus value is 350, Cl minus value lambda infinitive value is 76, Na plus lambda infinitive value is 50, and OH minus lambda infinitive value is 198. And Na plus also lambda infinitive value 50, and Cl minus is lambda infinitive value 76. So this is the value of the lambda infinity of the above ion. Now we will study about the conductance cell of the strong acid with the strong base neutralization titration. So first of all, you will be imagine the conductance cell. First of all, take a definite volume of an acid solution in the conductivity cell. Take a definite volume of an acid solution in the conductivity cell and cell connected with the conductometer. Cell is connected with the conductometer. First of all, we will take the beaker and we will be addition of the solution, and this is the acid solution. Mixture of HCl plus NaOH means sodium hydroxide and hydrochloric acid. And then after we will be dipped 2 electrodes in the solution, and 2 electrodes are is connected with the conductometer, and conductometer it will be connected to the rheostat. Now in the conductance cell, solution of hydrochloric acid, strong acid, and NaOH means sodium hydroxide strong base solution. So it will be ionization process, and H plus mobile ion and Cl minus mobile ion, Na plus mobile ion and OH minus mobile ion, it will be move in the solution, and it will be also connected to the conductometer. So take a definite volume of the acid solution in the conductivity cell and cell connect with the conductometer. First of all, we will take the beaker and addition the solution of strong acid and strong base, and then after we will be 2 electrode dipped into the acid solution, and 2 electrodes are connected with each other and connected to the conductometer, and then after we will be pass the current from the source, and it will be ionize the ion H plus and Cl minus and Na plus and OH minus, and these are the mobile ions, and it will be connected to the conductometer. And this figure is the conductance cell of the strong acid with the strong base of the neutralization titration. Now following equation we can calculate the concentration of the acid solution. So we will be calculate the concentration of the acid solution. This is the acid solution and we will be calculate the concentration. So therefore acid solution is equal to base solution, means N1V1 is equal to N2V2. This is our first equation. N1V1 is equal to N2V2. Then after N1 is title, and then after N2V2 upon V1. This is our equation number B. Equation number 2. Sorry, equation number 2 from the graph plot. This is the equation number 1 and this is the equation number 2. And equation number 2 will be calculated from the graph plot. So we will be see the figure of graph. Conductance versus volume of NaOH. Conductance versus volume of the NaOH. And the graph value and graph was curve. And here value of the H plus and Na plus ion, and here the value of the Na plus and OH minus. And that point is called end point or equivalent point. This point is called the end point or equivalent point. So graph will be draw the conductance versus volume of the NaOH, and we will calculate the V2 from the graph, and it is called end point or equivalent point. And this equation N1V1 is equal to N2V2 and N1 is equal to N2 into V2 upon V1, and it will be calculated from the graph plot. So this is the explanation of the neutralization process of the strong acid with the strong base. First of all, we will take the beaker and we will addition the solution of acid and strong base HCl plus NaOH, and then after we will dip the 2 electrode, and it is connected with each other and connected with the conductometer. Then after ionization process, 3 mobile ions H plus Cl minus and Na plus and OH minus, it will be connected with the conductometer, and the value of the lambda infinitive of the all mobile ions, and this is the equation of the concentration we will be calculate from the this equation 1 and 2, and this is the graph of the strong acid versus the strong base neutralization process. Graph will be drawn from conductance versus volume of NaOH, and V2 point is called endpoint or equivalent point. So this is the concept of the strong acid with the strong base neutralization titration, and we will discuss about the other three neutralization titration in the next lecture.